I know that some people have some concerns about mRNA vaccines, so I want to show you from a biochemist standpoint that mRNA is really nothing to be scared of. It's this natural thing our bodies use all the time. I'm not an immunologist, so I'm not going to try to get into that side of things. But the key thing to keep in mind on that front is that the goal of any vaccine for the coronavirus is to introduce your body to a form of the virus or a part of the virus in a way that's not going to make you sick. This way, your body can learn to be on alert for it in the future, such as with antibodies and T cell responses. So, I'm not going to try to get into that, but I am going to tell you more about mRNA. To the news and stuff, it makes it sound like it's this whole brand new thing, but it's really not. mRNA has been used by our cells forever, but the only new thing is that we have the technology to make it outside of the body in the lab and then introduce it into the body. Um, so basically, our cells hold the genetic recipes for proteins in the form of genes, which are stretches of DNA. In our cells, these genes are held in a membrane-bound compartment called the nucleus. And this is really great because it keeps all those original recipes safe. You don't want to mutate those because that could cause problems. But in order to actually make a protein based on those instructions, you're going to have to work with the recipe. But you don't want to work with the original recipe. So what the cells do is they make messenger RNA copies of the DNA gene. And then they send those copies out of the nucleus and into the cytoplasm, which is just the general cellular interior. And that's where the protein making complexes, the ribosomes, are. So this method is really great because not only does it protect those original copies, but it allows you to make lots and lots of copies of the recipe so that multiple ribosomes can be working on it at once. If you imagine like a restaurant, the restaurant isn't going to give their original copy to a chef to use. They'll get like spaghetti sauce or stuff all over it. Plus, you could only then give it to one chef at a time. But if you make copies of it, then you can give it to lots of chefs at once, and you protect that original copy. So that's the idea with mRNA. So in our cells, what happens is we have a gene that's in DNA. Then through a process of transcription, it gets made into a messenger RNA copy. And then that gets made into a protein in a process called translation. So RNA and DNA are really, really, really similar. So they're both these genetic languages that you can convert between. So you can make DNA copies of RNA and RNA copies of DNA and DNA copies of DNA. They just have a little different backbone and one different letter. So our cells have used mRNA forever, but the difference here is that we now have the technology to make synthetic mRNA. And by synthetic, we just mean that it's made in a lab. It's not any different than the mRNA that would normally be in your cells or that the virus would make. So it's the same mRNA. We're just not making it in the body. We're making it outside of the body and putting it inside the body. So, and then the other key technological innovation that is allowed for mRNA vaccines is the delivery method. So these mRNAs are going to get encapsulated in this oily lipid membrane that's going to help it get inside of the cells. And so we need this because if we look at that backbone of the RNA, we see it has this negative charge. And the lipids so lipids are just like fatty, oily things. And as you might have known, if you've ever tried to mix oil and water, they don't really mix. So the phospholipids surrounding your cells, they have parts that like water and parts that don't like water. And so the parts that like water, the phospholipid heads, are on the outside and on the inside. And so they form this like lipid sandwich, this bilayer. And these heads have negative charges. And negative charge, opposite like charges repel, so they're not going to want to get close. But if you surround the RNA in a positively charged lipid nanoparticle-y thing, you can neutralize the charge of the RNA, and you can surround it in this more lipidy coat that allows it to kind of slip into and merge with the cell membrane. So typically, I think it's thought to do this by endocytosis. So basically, it gets like swallowed in, and then once it's inside of the cell, the lipids that make up the lipid nanoparticle actually become positively charged once they're in those endosomes, because the endosomes acidify as part of the process of endosome maturation, and that acidification allows them to kind of nudge in between the endosomes walls and escape out. So let's look at what we want to put inside of what mRNA do we want to use. Well, if we look at what is in the SARS-CoV-2 virus, so it has this, basically, unlike our genome, so our DNA we have a DNA blueprint. They have an RNA blueprint. So they have this single strand of RNA, and then it's coated up in this nucleocapsid protein and surrounded by this membrane envelope. And then the thing that you've probably seen a lot is this spike protein. So that's the one that sticks out from the surface, kind of crown-like. And the virus uses this to dock onto cells. And once it docks onto cells, it merges, it gets either swallowed up or it can merge at the surface. But the basic idea is that that the spike protein is like the entry point. Once it's inside, then that RNA comes out, and the virus can start making mRNA copies from it, as well as full copies to make further viruses. The virus is making mRNA copies of the genes for proteins, including spike. And so our idea is to just introduce mRNA copies of the spike protein so that our cells can make the protein without having to actually be infected by the virus. Spike is the logical choice because, as we see, it's this one that is important for docking onto the cells and allowing the virus to get in. So if an antibody gets there first, so antibodies are those little proteins we talked about that can bind specifically. So if you have an antibody that binds to the spike protein, it can prevent it from docking onto our cells. And so we call antibodies that can do this neutralizing antibodies. And so these are like the golden ones that you want. So the mRNA vaccines, they only contain copies of the recipe for a single viral protein, so just the spike protein and not the whole virus. So all you're doing is putting copies of the recipe for the single protein. And these are temporary copies, just like all the mRNAs in our cells are temporary copies, which is one of the other benefits of mRNAs. So RNA is less stable than DNA, so it allows us to make temporary copies of things so we can regulate levels of the proteins that we make. So if we stick mRNAs from the coronavirus into our cells, those are going to be temporary as well. The way that this is done is through in vitro transcription. So basically, scientists can make large amounts of RNA outside of cells, taking the gene for the S protein and using a process called in vitro transcription. They can use this T7 DNA polymerase, which will make lots of copies of that RNA, and then they purify it out, surround it in these lipid nanoparticles, which can then let it get into the cells, and then the body can do all that antibody making and generation of T cell responses, which is more complicated, and so I'm not even going to try to explain all of that. But the basic idea is that you can get immunity to the virus without actually having to have the virus. And so this is just one way that you can introduce viral parts for the body to learn to recognize. So other methods include more traditional methods like using killed or inactivated viruses, weakened viruses. There's also the idea of using viral vectors, so using a different virus, such as an adenovirus, to deliver the S gene. And so all of these methods have the same goal, which is just to get your body on alert for the virus without actually having you get sick.